Genetic fingerprinting is a technique that can be used to analyse DNA fragments that have been cloned by PCR. You can look to see if there's genetic relationships or genetic variability within a population, or to see if you've got the same person or not within a population. What it uses is things called VNTRs, or variable number tandem repeats. These are found between genes, so not within a gene, but between genes. As you can see in the example above, we've got this repeated sequence. So AGT, CGG, TAAG, and that repeats 6 times in that individual, and it repeats 8 times in that individual. And these repeated sequences, the probability of 2 individuals having the same VNTRs, unless they're clones, is very, very low. So the more difference the length of these variable number tandem repeats, the greater the variation there is in the population. So how does the technique work? Well, first of all, you need a DNA sample. You need to extract the DNA sample. And then the DNA is cut using restriction enzymes. And it has to be the restriction enzymes have to be chosen so they do not cut within the tandem repeats, so they cut outside the tandem repeats. Then what happens is, and it doesn't tell you in the information here, these fragments would have to be cloned using PCR. So you've got lots of each copy of each fragment, and those fragments would vary in length depending on the size of your VNTRs. You then place those fragments into different wells in a gel, and an electric current is placed over the gel. And because of the phosphate in DNA being negative, phosphate ions being negatively charged, the DNA will move towards the positive end, and the smaller fragments will move further, move at a faster rate through the gel. Once you have got your banding pattern, it's then transferred to a nylon membrane. You then use a probe, so it could be radioactive probe, binding to the DNA fragment. And then once you've got your you've attached your probe, you can, if it's a radioactive probe, you can lay an X-ray film over the top, and it exposes the X-ray film, and you get a banding pattern where each VNTR was. So if this was the positive end of the gel, this would be the smallest band, and this would be the largest VNTR. So you can see we've got several different bands, each of different length, corresponding to the VNTRs of the length of the VNTRs of that person. More modern techniques might use fluorescent probes instead of relying on radioactive probes and X-ray film to expose and show where the bands are found. So what can gel electrophoresis be used for, genetic fingerprinting be used for? Well, forensic science is one that you should have heard about before. So here's the DNA extracted at the crime scene. It could be from a sample of blood that they found. And then we've got the victim. So the victim is there to eliminate their banding pattern from the results. And you've got 2 suspects. So which suspect's banding pattern matches? And you can see it's suspect 1 who've got exactly the same length VNTRs, exactly the same banding pattern, so the fragments are all exactly the same length. And the probability of a different person other than the person that committed the crime having the same VNTRs is very, very low. Another thing that genetic fingerprinting can be used for is medical diagnosis, to see if you've got perhaps a genetic disorder. So, in the normal gives this banding pattern, but the disease gives this banding pattern. And you can see the red lines are the ones that they are looking for, because that shows the differences. So what causes the difference in this example? So you can see the MST1 restriction sites, so this is a restriction enzyme. So in a normal, it would cut the DNA in 3 places, giving you a medium size length and a smaller size length. And we can see on the gel that this would correspond to this medium size, and this will correspond to this smaller size fragment that you would get. But in the disease there's a mutation, which means that the restriction site is no longer complementary to the shape of the active site of MST1. So MST1 cannot bind and cut the DNA at this point. So you've only got two restriction sites, meaning that you've got one large fragment of DNA when cut with that restriction enzyme. And you can see here that this piece of DNA hasn't moved as far, meaning it's longer. We've only got one band as well, showing this we've only got one, so that must be the disease. Another example that this can be used for is animal and plant breeding. It could be used for a couple of different reasons for this. I couldn't find any suitable images to use. So first of all, it can be used to see if the genetic variation within the population. So if you've got, for example, in a zoo, you might want to prevent inbreeding depression. So you're restricting the gene pool. You don't want relatives, necessarily too close relatives, to breed with each other. So you could use this technique to see if the organisms are far enough, have enough variation to breed. Or you could use it with selective breeding, a bit like the previous example, to see if there's a specific trait present in the animal or the plant before you breed them. So this could save time waiting to see if it shows in the phenotype, or it could increase the likelihood that the desirable trait will be found in the phenotype before, so you know which organisms to breed to start off with. When it comes to revision, please practice as many questions on this topic as possible, and you'll see the style of questions that you get. They're often quite application heavy.